talk not really about neurophysiology, but about some of the work we've done trying to predict seizure outcomes based on structural imaging. So I'll get into that a little bit more. But first, no real disclosures. Maybe relevant to this is a study we did for Medtronic looking at what happens to tissue during laser ablation. So I'm going to just give a quick overview of the talk. Originally, this was going to be a research talk, and then it kind of got extended. So instead of talking about some of the different research projects we have going on, I just thought I'd give one of our sort of lab research talks. Kind of easier to do for the length. So, but the study we were looking at outcomes after laser ablations for temporal lobe epilepsy. So I'll start with some background, some motivations for this, the types of things that we look for, so what we mean by structural connectivity, and then just go over the results of our study. I think as everybody in this audience would knows, the outcomes for temporal lobectomies for epilepsy are one of the few things that we have where we have randomized controlled trials in neurosurgery. So one of the classic papers, this BB paper in New England Journal, they did a randomized controlled trial comparing best medical management to anterior temporal lobectomy for temporal lobes epilepsy with about a 60% seizure freedom rate at a year. It's kind of, sort of standard neurosurgical lore at this point. So that's, it's about 60%. Again, as this audience knows, we've been doing laser ablations here at UW since about 2013. And so we looked at sort of 5 consecutive years of this. We saw our seizure freedom rates at 1 year were about 44%. So there's a significant disconnect between these two numbers. And we wanted to kind of look into this and potentially figure out some of the reasons. I think there's 2 potential sources. One is factors that have to do with the patient themselves. So clinical features of their epilepsy, perhaps like EEG, whether they have lesions on the MRI or other factors that could be looked at on imaging. And then, I do think there are probably surgical factors. So laser treatments ablate less brain. They can be at different locations. So potentially anatomic relationships for where you place laser catheters can affect your outcomes. Again, same thing goes for an anterior temporal lobectomy. So in terms of how much temporal lobe you take out, whether or not you use electrocorticography, those things can make a difference. Looking at our kind of results for the first about 100 patients or so, we looked at a couple of different sort of classic clinical features that might impact outcomes, including semiology, so whether or not what the seizure looks like kind of matches with a mesial temporal lobe onset, how often they have convulsions, what their EEG looks like, what their MRI look like, and basically using a stepwise regression kind of found that really the things that matter for our laser patients were whether or not they had mesial temporal sclerosis or whether or not they had interictal activity. So basically the things that you would expect, some of the things that you would expect indicating that maybe it isn't just mesial temporal lobe involved seem to herald a worse outcome. Some other folks, and this is just an example of what other people have looked at, kind of from a different standpoint. This study looked at 121 temporal lobe epilepsy patients, and just looked for potential connectivity data between regions to try and predict whether or not patients would be seizure-free. So they took 121 patients, which 80% were seizure-free, looked at diffusion-weighted imaging, and then kind of fed connectivity data into a neural net to try and figure out what, if there were any particular predictive features. And what they did, now I'll get into kind of what specifically these connectivity measures are, but they found they were able to predict whether or not people would be seizure-free in about 2/3 of the time using a metric called betweenness connectivity. So patients who had less of this particular feature were less likely to be feature-free or to be seizure-free. Interestingly, this seems to be independent of what kind of surgery was done. So laser ablation versus an anterior temporal lobectomy didn't make a difference. Whether or not the patient had a lesion, so like mesial temporal sclerosis, did not seem to make a difference, and no other clinical or demographic features were predictive. So things that you would normally look at from a clinical standpoint, apparently this imaging finding kind of trumped all of that. So this is another kind of approach to looking at patient factors, is to say, look, we're just going to look at their brain, features of the brain, and see if we can tell how people are going to do. So this slide just kind of summarizes that. So we may be able to predict how these things will, how outcomes will go without actually knowing much else about the patient. In kind of contrast to this, I think a lot of us who do this do believe that what kind of surgery you do and how well you do it makes a difference. And so there's a multi-centered study of which we contributed a significant number of patients, but we looked at about 230 patients who underwent a laser ablation, kind of looking at where the ablation was and whether or not the location of the ablation had to do with seizure freedom. And basically, roughly about 55 to 60% of patients are seizure free at a year. And the key thing is, within this group, a very careful normalization of laser lesions was done and kind of imposed onto a high resolution standardized map. So this brain behind this is kind of, the 200 or so people's brains co-registered to each other, meaning you kind of smash them all together, but line things up as best you can, including the lesion. And basically what we see is as you look at what parts of the brain were ablated, the positive predictive value map here shows the more, basically, if you ablate this area, you have a higher chance of having seizure freedom. You notice it's like kind of the medial portion here of the uncus, it's more anterior. And these are just some other views of this showing kind of how likely it is for you to be seizure free if you had a particular voxel ablated. So important things here were just that, again, this anterior, medial, and inferior area, basically as much amygdala and uncus as you can get. And as you go back in the hippocampus, there's just less value in ablating sort of the tail. And we'll get back to that too in a little bit. One other paper of interest that looked at surgical factors, looked at anterior temporal lobectomies, and kind of what changed in terms of structural connectivity after doing the resection. And so that first paper I showed you said, look, this is just brains and the connectivity within the brain. This paper looked at that same connectivity, but kind of simulated changes after a resection. So they took whatever connections went through the part of the brain that was removed during an anterior temporal lobe and said, okay, well, we'll recalculate metrics and see what happens with predictions. And basically, you can also use this type of data to predict surgical outcomes. So it's not just a patient factor, but probably something to do with what you've done during surgery that can be predicted. So again, this slide just summarizes that. We can predict outcomes just based on where the lesion is itself. And then even at least after a temporal lobectomy, you can predict changes in structural connectivity that are related to outcomes. And the point of the rest of this talk is to kind of look at whether or not we can do the same thing for laser ablations. If there's a relationship between anatomic predictors of success, so that medial ablation and structural connectivity, which again, we think has something to do with how well people do after surgery. So to talk about this connectivity that I've mentioned, what we're kind of looking at is diffusion weighted imaging. It's a pretty common thing that we do now. If you look at any of our clinical MRIs, if you have an ADC, that's a measure of diffusion. There are, and any of Christine McDonald's work with TBI looking at white matter integrity, so FA, mean diffusivity, those other metrics, those are important in all sorts of disease states. We're not really looking at that today. We're looking more at sort of reconstructions of white matter tracts or tractography. And the thought is we can use the number of tracts generated by some of these algorithms to reflect how connected brain regions are, not necessarily as a number of axons, but that's a good sort of surrogate way to think about it, I think. So in terms of how this type of imaging works, we can use MRI to measure water diffusion within the brain. And so normal diffusion without any sort of imposed structure, you're going to see sort of water move in a sphere in 3 dimensions. If there's structure in the scan that you're looking at, so if there's a brain there and the brain has white matter tracts, you can see effects on that diffusion because of that structure. With again, sort of the imaging here, or the diagrams here, if you have a group of axons, water tends to travel more easily along in the direction of those axons rather than across it. And basically, you can acquire the diffusion imaging in different directions to try and kind of reconstruct how water is moving in those directions. So just at the bare minimum, you need 6 directions to do any sort of reconstructions. That kind of makes sense, right? So up, down, left, right, front, back. The more directions you have, the more information you can get out of these scans, but obviously the more time it takes. And so our scans are usually about, at least for clinical use, about 20 or 32 directions. But what you can do is with those different directions, build models for each voxel in a scan. So each little pixel, you can say, there's 32 directions in our scan. For each voxel, there's a certain tendency for the water to be moving in certain directions. And you can do this in different ways. So the simple way to do it is just you come up with just an ellipse, basically, like this is the overall tendency. Yes, you have 32 directions, but I'm going to look at this sort of big oval. Another way to look at it is to look at a distribution of directions. And so you get this sort of lobed model that you can line up if you're going to do something like tractography. And basically try and model what happens with white matter. And so this sort of big distinction between different methods is a deterministic versus a probabilistic methodology. Deterministic is sort of, it's easier in some ways. And again, you're using that sort of single direction model. I know it's not a single direction, but basically, if you line up these little spheroids or ellipses, see which one, from one voxel to the other, say line them up, say they're pointing to each other, they're therefore connected, and you kind of make a decision for each voxel that it's connected to another one or not. You kind of get one answer. So a lot of the tractography solutions that people are presenting for clinical use, like QuickTome and things like that, basically they're simplifying the 32 directions into like one direction. Sort of a more accurate and interesting way to do it is with probabilistic tractography, where you allow some wiggle room for where which connections go to each other. and you do it multiple times, and take that data, and sort of this image here kind of shows that you can actually show crossing fibers and things like that. So, one voxel can then have 32 or more different connections to nearby voxels. You can look at tracks that overlap, and I think it's a more accurate way representation of how the brain works, but does take a lot longer. Again, in terms of how people use this data, one common method is to take the brain and kind of label different regions. So up here you have red for sensory, blue for motor. There's different atlases that are sort of generally accepted that you can automatically label, and once you do that, you can then quantify number of connections, number of tracks, number of simulated white matter tracks between each of these different regions. And we call this a connectome. And basically you can represent that as a matrix or a graph. So to look at some of the graph methodologies that we used, each of those regions of interest from an atlas can be looked at as nodes, and then connections between nodes are called edges. And there's useful reasons to kind of think of brains as graphs. So different regions like amygdala, hippocampus, how connected they are to each other, you can kind of quantitatively represent it this way. So just looking at this dummy graph over on the side here, this has 8 nodes, and then the lines that are red here just represent edges. They kind of represent connections. You can consider it like sort of ones and zeros, like yes, there is a connection, or zero, there is not. Or you can sort of weight these edges by looking at streamlines generated through tractography. So it's sort of a measure of how connected these areas are, and we call that a connection strength. There's different other characteristics that you can look at. And so these network metrics can describe either nodes themselves, like nodes, so different regions, or edges. And so for example, one measure of node strength is how many other areas a particular node interacts with. So again, here's an example. There's 4 edges leaving this node, so the node strength is 4. There's 2 from this other node, and so one is more strongly connected than the other. And you can do that with either just the number of connections or the number of streamlines generated by your diffusion tractography. And that's sort of description of a single region. You can look at sort of neighborhoods as well. So different regions that are different nodes that are next to each other will relate to others in the area, and you can describe certain network metrics that way. So if you look at node A here, there's 3 neighbors. Node B has 2. Okay, so that's a node strength 3. So the node values particular to A or B. We can also look at how connected their neighbors are. So for node A, there's its 3 neighbors have 3 possible connections to each other. Only 2 of the 3 actually exist. So we call this the clustering coefficient. So how connected are our neighbors' or our nodes' neighbors. So this clustering coefficient is 2/3. If you look at node B, there are fewer connections, but the neighbors are very tightly connected. There's only 1 possible connection, and it's there. So the clustering coefficient is 1. And this is just a way to look at not just an isolated node, but how its neighbors interact. Another measure of interest when we look at this is something called centrality. And to a certain extent, you can think of it as how important a node is to the structure of the graph. In other words, if you take out a particular node, does the graph fall apart? So that example here is if you take out the node in blue, now you have 2 unconnected regions. So there's a high betweenness connectivity for that one particular node. This is another example. where if you remove this blue node, all of a sudden now you have, again, sort of two separate graph regions. On the other hand, this node, if you took it out, you still have a pretty unchanged structure for the rest of it. I.e., if you wanted to walk from one point to another on this graph, you can still do so, having to stay on edges that exist, so staying on lines that are there, you can still get from one end to the other. Whereas in this case, you would not be able to get from one place to another if you remove this node. So these measures, again, just to get back to why we're talking about this in the first place. Basically, the two measures I've described have just been shown to be important for various different types of networks and connections. So like social media, between this connectivity is indicative of certain strengths of networks. But in some other studies, predicting seizure outcomes, a lot of those two measures in particular have been of interest. But what we want to do here is employ this change in connectivity approach I mentioned before. So for each of our patients, we want to take a preoperative structural connectivity measure and sort of simulate what happens when you cause your laser lesion. And how we do that is just we generate our connectome as I described before, and then just say, look, there's this area here where the laser is. Any connection that travels through that area, we're not going to count it when we're doing our streamline counts, and basically look at the percent change that's a result of doing the lesion. And then basically use some machine learning techniques to identify which of those changes are predictive of outcome. So it's important. This is not like a study where we did a DWI before and after the surgery. This is us saying in an ideal ablation, this is what happened, and this is what changed. And kind of you can see the workflow over on the right there. But we looked at 24 patients with mesial temporal lobe epilepsy. They all had to have 1-year follow-up. We looked at clinical features, things I talked about earlier that might predict outcomes, and basically didn't see anything that was predictive. And again, to do this, we quantified connectivity using a fairly standard atlas. It's the DK Desiccant-Kiliani. It's an atlas that basically parcelates the brain into different regions, and looked at the percent change. And so each of these connections across both hemispheres, basically we made them all left temporal lobectomies or left temporal lasers, as we can have a slightly more symmetric comparison. But when we looked at which connections changed, you can see there's a whole lot of different number of potential interactions. So given that the atlas has 84 regions, you can kind of do the math. But if we were to try to look at how each of these 3,400 connections impacted outcomes, it would be pretty hard. You'd think you'd have a lot of false positives, a lot of type 1 error from doing this. If you look at the node-based metrics, just the node-based metrics, if we had 84 nodes for each patient, again, with 24 patients, you're probably going to have some difficulty having too many potential predictive features to look at. And so what we ended up doing are some of these they're kind of standard now, but machine learning approaches to try and cut down the feature numbers and really pick out which ones are important before trying to do predictions. So technique in particular, we use logistic regression with an elastic net regularization, and then put the output from that into a support vector machine. And then for each set of parameters for the elastic net, kind of looked at which gave the best predictions. So basically, we identify features that seem to be predictive out of the 3,400, test whether or not they are predictive, and then using subsets of data kind of cross validate. So ultimately, you get an area under the ROC curve. We use the Matthews correlation coefficient, which is it's a nice parameterized test of whether or not your algorithm does better at predicting outcomes than chance. And then we also did surrogate predictions. So if you basically create fake data using your own data and kind of destroy correlations between within that data, you can set sort of a baseline for if you were just guessing with your own data, what would you get? And so, again, in terms of looking at the different types of network measures, if you look at node strength, so again, that measure of how connected a certain node is, the entorhinal cortex, fusiform gyrus, parahippocampal gyrus, temporal pole, hippocampus, and amygdala change the most. And that kind of makes sense, right? If you think about where the lesion is, that's where the laser goes. But using this regression followed by a machine learning or support vector machine, we found that how much you change the connectivity of entorhinal cortex is the most predictive of whether or not people will be seizure free. And this was significant with respect to doing better than chance, and the accuracy was modest, but the AUC is 0.66. You can see similar sort of changes if you use clustering coefficient. And again, if you look at what happens after the ablation, those 6 regions kind of do change a lot, but the ones that help predict outcomes are the entorhinal cortex, parahippocampal gyrus, and temporal pole. And so, again, kind of local to the lesion, but it may be not so surprising given percent changes in the area close to the laser. Looking at between the centrality, so this particular metric, even if you make a local change, you can a change that's local because you're looking at how it affects features across an entire graph. You'll see changes even on both sides of the brain. But again, with our algorithm, basically picked out entorhinal cortex and temporal pole as regions that are important. And you'll notice the area under the curve, they're all pretty modestly better than if you just were picking things by chance in terms of guessing whether a patient would be seizure free or not. That said, if you look just at connection strength, so again, that first graph I showed you, like these are all of the different connections, and what happens, you can see our algorithm picked out sort of 5 regions that where it changes, where you can kind of draw a line between seizure-free and non-seizure-free patients, and do a pretty good job guessing of what happens. Our area under the curve here is 0.85, which so it's just does a better job overall using these 5 features rather than trying to use a single regional change. So the overall accuracy using this is about 88%. The connections are primarily local to the lesion, so ipsilateral mesial temporal lobe. The connection strength that the connections that are kind of best predictive, 3 of them involve entorhinal cortex. So entorhinal cortex, insula, hippocampus, amygdala. Okay, probably not surprising. Fusiform gyrus to caudate nucleus was also predictive, but sort of in an inverse way. So the more you ablated this particular connection, the less likely you are to be seizure free, which I think we get to a reasoning for this soon. And then temporal pole hippocampus was also predictive. So it seems like if you get more of these 5 particular connections, you have a better chance of doing what you came to do. So just in terms of what this means, that's important with any of these machine learning algorithms to say, like, these are not like statistically significant changes. So if you did like t-tests or something like that, you wouldn't find it a significant outcome. But all this is kind of derived from the machine learning process. So it's just basically like, look, if you took subsets of this data or changed little aspects of this data and ran the same algorithms over and over, you get the same answer. And so that's sort of the validity. And it's just kind of important to keep that in mind, that for any machine learning algorithm, that's how these accuracy numbers are generated. And then you kind of compare that to a chance distribution. So again, the Matthew correlation coefficient, it's just a good way to measure accuracy of these sort of, we call these supervised learning algorithms. But when we do that, it's nice to have a parameterized test, so you don't have to permute data a lot, but we did both. So these connections that change, that's all well and good. We wanted to see whether or not these connections lived in a particular area of the brain, basically. So with the previous paper on looking at medial and anterior ablations, is that reflected in connection strengths and what happens when we look at structural connectivity? So basically, when you do these lasers, the catheters can be in different positions. There's going to be variability, but with this way of simulating changes in connections, you can kind of artificially manipulate the lesion and see what happens to the connectome. So basically, you can look at just the easiest thing is like if you look in 8 different directions and expand your simulated lesion in that direction, how does it change the rest of the connectivity? And so basically we did that. And if you look at dilating the mask, so making the simulated laser area bigger, as in B above, you see that as you sort of increase its size in the medial, anterior, and inferior direction, you further decrease connection strength in entorhinal cortex to insula, to hippocampus to amygdala, and basically the connections that are predictive. If you dilate laterally and superiorly, so kind of at the tail of the hippocampus, you start to decrease connection strength more in the fusiform gyrus and caudate to caudate connection, and basically thus kind of recapitulating the model prediction. So in terms of whether somebody be seizure free, as you increase the medial, anterior, and inferior portion of your laser, you get more seizure free predictions. So this kind of can be, when we do this, we do it in every individual's particular DWI space. And so you get individual predictions, which is good. Kind of knowing where each of these things are across patients would be a good idea as well, just to help think about things. And you can do this, again, by transforming things into a standard space. So looking at our actual laser ablations, if you put them all into the usual, this is an MNI brain, but it's kind of a standard brain. You can transform other individual brains and kind of shift it and match it up to these standardized brains. And when we do that, this is again just using the actual lesions. If you look at this bottom row here, more red means more likely to be seizure free. And so as you look sort of inferior and medial, you have an increased chance of that. That's with the real data. If you kind of look at the data that we use to impact connections, you can see sort of, instead of looking at each individual person's, like on average across the patients, if you did not have, if your laser lesion did not cover a certain area, how likely were you to be seizure free? If it did, how likely were you to get a seizure free prediction if you kind of faked a new laser? And this top row here is kind of what you see in terms of the number of seizure-free predictions. So this is just sort of another way to look at the connections that you influence. So as you dilate your lesion, like if you didn't have this area burned initially, and then you, in our simulated laser ablation study, if you simulate that it was, and you look at the connections that change, the likelihood of your seizure prediction, your seizure outcome prediction going from negative to positive, basically is very high if you are looking in that medial and inferior area and anterior area. So basically, this is just a sort of anatomic confirmation that these more medial ablation locations lead to a higher seizure freedom rate. This is probably not surprising, given that this is a subset of the 234 patients in the blue cohort in that paper I showed. Other important things are that looking at the connectomic changes, so what, not just where the lesion is, but which connections are affected, can predict seizure outcome. So between this connectivity and clustering coefficient, things that people like using, they work. But if you just, at least for this particular use, the connection strength alone just kind of had higher predictive value. And these were local to the mesial temporal lobe, which is, again, if you think of epilepsy as a network disease, this is a little bit surprising, but it also kind of makes sense. Some things that need to be addressed, or I think are problematic with this approach. Basically, we've just used preoperative data, so we don't know if when we say changes in structural connectivity, they're not actual changes, they're not measured, I guess I should say. We also have ignored left and right. Here we kind of made everybody left, and we do know that right-sided temporal lobe epilepsy and left-sided temporal lobe epilepsy are different. But again, in order to get sort of numbers, we ignored that. This approach, looking at a change in connectivity, is really useful if you have an existing lesion. So, in terms of whether or not this particular approach is clinically relevant, to a certain extent, yes. So what you do is, if you're looking at a lesion or you're looking at a patient and they've already had surgery, and let's say they fail, so they start having epilepsy after you've done the laser, you could run this algorithm and say, okay, well, would this algorithm have predicted seizure freedom? And if it didn't, then it probably wasn't a good laser. So it's useful if you have an existing lesion. On the other hand, we don't have good ways of using this to help plan surgery before you do the laser. So one, we don't really have a good way of simulating laser lesions accurately. So the second is this model: the bigger you make your lesion, the higher your seizure freedom rate. So if you just keep increasing the size of your lesion to simulate in the beginning, it's just going to tell you to laser the whole temporal lobe, which would then be the same as a temporal lobectomy. The 24 patients is a pretty small number. We do have additional patients at this point over the last 3 years that we could add, and so we're kind of in the process. In terms of what we'd like to do next, what I'd like to do next, basically, there's that funny thing where some models, I showed one paper earlier with like a 66% accuracy, but there are some now that people are able to predict with like 97% accuracy whether or not you'll be seizure free, just based on MRI features, completely ignoring what kind of surgery you had. And to me, that's like, okay, I think they're overfitting, but that's it's still I think it still does tell you that there's a sort of a fingerprint of patients that are going to do better. And looking more into that is important, while taking into account that surgery, I think, again, you can do a good or bad job doing surgery. Again, we'd also like to look at individual laser trajectories. we have ongoing collaborations with people who are doing, simulation of lesions, automated planning of laser placements and things like that. But ultimately, what we'd like to do is be able to predict who should get maybe an ATL instead of a laser, save somebody a surgery, or figure out, look, even if you did do a bigger surgery, an anterior temporal lobectomy, there's still 40% of patients who won't be seizure free. Should we skip to something like RNS or VNS or DBS, where you're not going to cause the side effects of this type of resection without the benefit, and yeah, you don't get as good a result with neuromodulation necessarily, but you also don't have the deficits that you cause. And so trying to use some of this data to predict that would be helpful. And just a small number of the people who helped with this, basically the author list on that paper,